Good morning, everyone. It's 11 o'clock, and so let's start the summit. I'm the first speaker, so to paraphrase the iconic Austin Powers, please allow myself to introduce myself. I am Dr. Julie Irvin. I'm an evolutionary biologist, and I've been working on lanternfly since it first arrived, since it was first detected in the United States in 2014. And I was working in North Carolina at the time. And after that, in 2016, I had the opportunity to come to Penn State, where I now work. And so I've essentially had a front row seat of lanternfly for these past 11 generations. And why I say 11 generations is that, many of you are very familiar with spotted lanternfly. You've come to these summits in the past. Some of you might be new, but, to remind everyone, lanternfly only goes through one generation per year. And so what I wanted to do before I gave you a bit of an update on feeding behavior and phenology work that I'm doing, I wanted to, in lieu of spotted lanternfly 101, go through essentially what I see as some of the milestones, like the research milestones that have influenced or shifted how we think about spotted lanternfly management and all of the complexities associated with spotted lanternfly management. So back in the beginning, if I could find, here we go. Lanternfly was first detected in Berks County, Pennsylvania, September 22nd, 2014. Within 2 weeks of that detection, USDA APHIS organized a technical working group of scientists from around the country. Basically that first spring, a group of us came. We were hosted by APHIS and Pennsylvania Department of Agriculture, and we were taken to the area where lanternfly was first detected. And so you can see we photographed egg masses, and we actually saw the first recorded hatch that first year when it was known that they were here. And so at that time, if we think about milestones, how lanternfly treatment was thought about, it was actually very ingenious. Oh, sorry, I got ahead of myself here. Basically, what we knew at that time point, if we're talking about milestones, we knew what spotted lanternfly did as an invasive in South Korea. And so from that, our knowledge basically was that spotted lanternfly feeds on over 70 species of plants. We know that's, an even higher total now on record, over 100. But tree of heaven, Ailanthus altissima, is one of its favorite hosts. We also knew from reports from South Korea that feeding and honeydew deposition, so that's its excrement, which is a substrate often for sooty mold growth, those pose a risk to grapevines. That's the biggest threat, but also to a wide range of products and associated industries. So apple, stone fruit, ornamental trees, woody trees, cherry, lilac, and a large range of specialty crops, which really was concerning, particularly in a state like Pennsylvania, where we have a lot of fruit, grape, and vegetable production, but also a lot of timber. Timber is a very important economic industry. And even taking a quote from Dara's article, he mentioned that SLF could potentially impact industries ranging from lumber to wine. Okay, so what happened next? Well, basically, using that piece of knowledge about spotted lanternflies' preference for tree of heaven, Pennsylvania Department of Ag and APHIS, the treatment protocol was to focus on Ailanthus. And so basically, Ailanthus were established as trap trees. So the main management approach was to remove 90% of the existing Ailanthus trees, leaving only the males, so you don't have trees that produce seeds, and treat the remaining with dinotefuran insecticide, so a neonicotinoid that would get injected or uptaken into the trunk of the tree. And this is pretty ingenious because basically we're concerned, and we continue to be concerned, about insecticides potentially killing off or injuring non-target pests. And by focusing treatment on tree of heaven, which itself is an introduced invasive and generally isn't used by pollinators or other beneficial or other arthropods. So that was a way to be selective for killing lanternfly, but also it was look at these numbers. You can see the wing spread. You can see injecting a tree, as we've found in a subsequent study that Brian Walsh conducted. In 7 days, one tree that he injected, he went and counted all the dead lanternfly, and it killed over 12,000 lanternfly in 7 days. And so this was, incredibly effective at killing large numbers of lanternfly, but we know, because we're here today, that alone wasn't enough. And so lessons learned from that, first big milestone, treating tree of heaven isn't enough. It's just going to take time. It's impractical. It's expensive, especially in the early days, trying when lanternfly wasn't really recognized right off the bat as dangerous of a pest that it is. We're just behind the eight ball in a sense, but also SLF is highly mobile and feeds on other hosts besides tree of heaven. So management strategies changed really immediately, and government agencies now use knockdown contact applications, oftentimes bifenthrin, and egg mass treatments are also really important. And so this comes to the second milestone. And again, this is from my perspective. Okay. There's certainly, I don't have time to go through all the really cool research studies and great findings that have been done. That's why you're going to hear a number of those today and, on Thursday as well. But this focus on egg masses was really critical, because if you think about spotted lanternfly's life cycle, eggs are laid October, November, and then lanternfly remains in that dormant egg stage from November till April and into May. So it's essentially half of the year spotted lanternfly is an egg mass. And so thinking about that seems like, wow, if we could really focus on egg masses, that would be a great target, because they spend half of their life in that stage. And we know, though, that from trials that were conducted, ovicides have variable efficacy. The best you're really going to get might be killing 75% of eggs. And they're most effective within a narrow window. It's still, 3 months, but you need to provide high enough volumes to provide good coverage. But the study that kind of really hit the nail on the head here was led by Joe Keller, who was at Penn State. He's now at Temple University. And he took advantage of the fact that a number of parks and recreational areas in eastern Pennsylvania that had lanternfly wanted to take down their tree of heaven just to, reduce numbers in these recreational areas. And so he contacted folks at those parks departments. And when they were felling their trees, he got them, or, got Brian Walsh on our team, to cut the down tree into 3-meter segments to allow us to count how many egg masses were in each 3-meter segment from the ground on up. So answering the question, how, where are most of the egg masses laid, in terms of the vertical distribution. And what he found here, as you can see in this histogram, most egg masses were over 6 meters above the ground, so not really reachable. And so basically what we learned here is, a lesson that keeps on coming up. There's no silver bullet, okay? You can't just focus on egg masses, and not that that's what we were trying to do at the time, but this hit home the message that effective management requires multiple tools targeting SLF across all of its life stages. So that focus on egg masses is just one component. But, and here's where some of the lanternfly complexity comes in, it's not that egg masses aren't really important. And so a focus on egg masses can and has yielded some really powerful tools for reducing risk of SLF spread, for example, on nursery stock. And so we see a number of states, Pennsylvania and North Carolina, have trained sniffer dogs to detect the scent of lanternfly egg masses. And this is a very, very powerful detection tool. There are other tools in the pike, fumigants being worked on by USDA ARS, other methods of detection for egg masses. So this is certainly an important part of lanternfly management. Okay, next, this tree of heaven question. And this, does lanternfly require tree of heaven? Because we know in multiple studies, when you cage lanternfly on host plants, grapevine alone, grapevine with ailanthus, ailanthus alone, or a number of different different hosts, we know that in those cage studies, if lanternfly is given ailanthus, it improves their fitness, it increases their survival, etc. But kind of what we suspected and what Kelly Hoover's group showed in caging lanternfly in very large enclosures of trees that were actually in the ground. No, lanternfly can complete its development without access to Tree of Heaven. It does not require it. Okay, so what does that mean? Because when, based on a lot of these cage studies, things we hear in the press, we get questions. So does that, all we have to do is remove Tree of Heaven and we're fine? Well, no, it's not that simple. But does that mean that Tree of Heaven isn't important in terms of lanternfly management? No, of course it is. Okay. Again, no silver bullet. Even if you could remove all the ailanthus, that's not going to do it. But certainly ailanthus is important when, and it helps lanternfly establish in new areas. And so what about removing Tree of Heaven? Well, there's a number of biological control agents available or that have been studied and worked on for Tree of Heaven. Scott Salem talked about a weevil last year at the summit. If you missed that, that's in the recording. But there's a Verticillium wilt, Verticillium non-alfalfae, that has been studied and is available for limited scientific testing. And basically, as part of Tracy Leskey's USDA ARS funded area-wide project, Scott Salem and Carrie Fearer are working on that. And so this could be a cost-effective way to test what Tree of Heaven removal does, particularly around vineyards in the case of this area-wide study. So this is promising. But again, these issues with lanternfly are complex. In 2017, what we saw is that the SLF population seemed to boom. So you've probably seen this video before if you've ever seen me talk or you've seen it online. We see massive numbers of adults in vineyards. In that year, we also saw them moving on to other hosts en masse. This was in that same region. But subsequent studies since that time have led by, in the case of apple and peach, apple and stone fruit, led by Tracy Leskey's group, and on ornamentals led by Kelly Hoover's group, where they caged lanternfly on these respective hosts and kind of force-fed the lanternfly just those hosts, and then looked at what the physiological impact was on those trees. And what they found was lanternfly feeding itself isn't all that damaging. For ornamentals, it is a stressor, and it could be complicated if you have other stressors involved. But basically, what we found was a relief that, these other crops and essentially timber trees weren't at risk for severe damage from lanternfly as we had expected. But what we found is that, yes, it is, lanternfly feeding is really, really damaging to grapes and to Tree of Heaven. And so particularly the problem here is late season movement. This is a vineyard. You can see those are not grapes on the ground. Those are lanternfly that have been hit with a contact insecticide just prior to harvest. It knocked down numbers, but lanternfly, you can see them up here, still flying in numbers. And so Heather Leach led some studies. We see that this influx in vineyards in the late season, this late season movement, is what's really problematic for grapes, but it's also problematic in other habitats. And so there was a study led by Dennis Calvin here at Penn State, where we basically wanted to record this late season movement, in this case onto red maple, in order to develop degree day models to predict the timing of that influx. And so what you see here is in Wyomissing, Pennsylvania, outside of Reading, a suburban development. It's not an experimental plot for red maple, but it's kind of as close as you could get, in that it's all these little dots are red maples that were planted at the same time when the development was built, and they're all generally small. And so there were 19 red maple trees and 1 pin oak. And so basically we collected and sampled out spotted lanternfly for 2 seasons from just those trees in order to develop this degree day model. And so the way that looked, you can see here's one of these trees. There were sticky bands on the trunk, and they were collected whenever they were filled and they were counted. And then in terms of egg masses, because again, these were smaller trees, the egg masses were counted as high as could be seen from the tree, which was pretty inclusive. And so from that, basically, this is another game changer, at least to me, in terms of thinking about the complexity of lanternfly. And what you can see is of these 20 trees, this is the mean number of insects per life stage per tree. So for egg stages, this is counting out basically estimating 37 eggs per egg mass. That gave us a starting point of about almost just short of 500 spotted lanternfly per tree. And go figure, if you're trapping them out on sticky bands every week, what you see is you get this progressive drop, but boom, late season movement, you see a big influx of adults migrating in. And then when we go to count, okay, how many lanternfly does that mean in terms of then when they lay eggs? And we have to change the scale because you have the population getting that much bigger in that 1 year. And so what those numbers look like is these are lanternfly numbers out of those 20 trees in those 2 years. So we know red maple is a good host, but basically what this is showing us is that, oh my gosh, lanternfly really is highly mobile and it's constantly moving and changing hosts and feeding. And so the question in thinking about lanternfly as a landscape pest, and I'm kind of convinced if we could answer this question really well, we'd have a leg up on management in terms of where we are. What is that physical range that one lanternfly generation traverses? And so we know in the case of this Wyomissing study, this is an area rife with tree of heaven. And so they came in from that, but they didn't just come from there. And so moving on from that, going to the degree day model, this is what kind of inspired the work that I'm doing. What this showed is that basically looking at those accumulations of insects, particularly the adults, we collected them into ethanol. And Dennis built a degree day model that allowed us to predict egg mass deposition, but also that influx of males and females into this habitat at the end of the season. And so basically what we know, this is looking at a growing degree map. One of the interpretations of the study is, okay, how many growing degrees does it take for a female to reach reproductive maturation and lay eggs? Because if we look at how many growing degree days, this is a base that we use for spotted lanternfly development, what does that look like in terms of temperature distribution across, most of its range here in the east? And so we can see that basically lanternflies are going to mature earlier as they move south, which makes sense. And so Melody Keena's group has looked in terms of what does that mean for when they lay eggs, and that's its own other story. I'm interested in that late season movement. They're moving on to other hosts, grapevine and other hosts in particular, and they're starting to feed heavily because they need to feed heavily to become reproductively mature. They're putting on the feed bag, right? And so here, what I'm wondering is, are lanternfly populations, as they push south, going to feed more heavily? Are they going to basically start their heavy feeding a bit earlier and therefore feed longer in warmer regions? So are we going to see a greater impact in terms of resource depletion of various lanternfly hosts as lanternfly moves south in its range? And alternatively, is there going to be essentially lighter pressure, less heavy feeding in more northern regions? And so if we look at the current distribution of lanternfly, what I've been working on is collecting lanternfly from sites across this range. And so basically this is just showing you in 2024, I tried to do as close in time as I could, collecting at what was then the southern end of the range in Kernersville, North Carolina, Nashville, Tennessee, and then up here in Lewistown, Pennsylvania, and at the northernmost part of its range, Rochester. And you'll notice I couldn't collect adults until August in either of those sites because they didn't develop into adults until then. And then I recorded their live mass, and that's kind of my index of how heavily they were feeding, but also I'm dissecting each insect to look internally at the female maturation level. And so what this is kind of showing you here is, okay, again, think of these masses as a proxy for how much they're feeding. And so what we can see in those first three collections in North Carolina and Tennessee, they get increasingly larger over time. Putting in the masses of the Pennsylvania in blue and Rochester in green individuals, you can see this is what we expect. This suggests that their heavy feeding is starting a little bit later. But then, so this is kind of what we would expect, consistent with that hypothesis of heavier feeding in the South. But I was really surprised when I went back in September and got this unusual result for Rochester. And so the Pennsylvania population was still relatively small, but with a lot of variation. Rochester really those females were very heavy. And so, okay, does that mean that Rochester had a particularly warm year? Not really. If you look at growing degree days, not at all. They just basically were able to feed more heavily and kind of reach reproductive maturation quicker. And so kind of in conclusion, basically I had another collection site and we could see, the South certainly kept feeding heavily and, but again, that Rochester was unusual. So in terms of, yes, we should be a little bit worried that in warmer regions, heavy feeding could occur earlier and occur longer. But then this is a real surprise. In cooler regions, we didn't expect that we would see such large individuals at all. And these New York results weren't predicted by any models. And so looking at the lessons learned here, we really need to take a multi-method approach with lanternfly. We have the lab studies and the semi-field studies where we control, for example, what they eat and whatnot, super important, but we really need to ground truth that in actual wild populations in real places, because this is a super complex pest. And so where I want to leave us with is if we look at this current lanternfly distribution, we have it now in North Carolina, Atlanta, South Carolina, and these are just full counties. If you look at where lanternfly is within each of these counties, they're certainly not equally distributed. And so in some places, even where you have infestations like pushing up here in the lower Hudson River Valley, lanternfly have only just begun to get into vineyards in that region. And so if we look at this USDA Census of Agriculture map to see where grapes are produced, the darker shaded counties have more grape production. What we see is that lanternfly is just in the crosshairs of two really important Concord grape producing regions, the southwestern Michigan and the Erie Finger Lakes. And certainly here they're pushing into higher value wine producing regions in the east. And so obviously the West Coast is really important. And so part of our last take-home lesson is that regions need to be prepared for the arrival of lanternfly. We're doing something in particular with Josh Mills of Washington Department of State. He's interested in figuring out what lanternfly might feed on to traverse through the state if there were an introduction to get to vineyards. So we're working on a project where he sent us common corridor plants to Pennsylvania. My technician Tess has been rearing those. So this is what they look like when they're dug up for Washington State. So we can test them on lanternfly, but also we're trying to develop a protocol that could be more generalizable beyond lanternfly to enhance early detection and help learn how to rear native hosts for looking at basically non-target species that need to be tested for biological control. And so finally, the most important thing is we need input from regional stakeholders. That's why we're here with the summit. We've had panels in 2024 and 2025, and I'm really looking forward to the panel on Thursday with grape industry, and because we really need these cross-silo conversations. So with that, I will take my leave. I am right at 11:25, and thank you very much. I'm going to stop sharing, and I will take any questions in the Q&A. Thank you.